Program Manager for the 3Rs Collaborative. So we're happy to have you here at the MPS Lung Workshop today. So first, just a quick disclaimer. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3Rs Collaborative nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. So first, I'll just do a quick introduction to the 3RC, and then we'll have David from IQ do a quick introduction. So the 3Rs Collaborative's MPS initiative, our goal is to collaborate to accelerate adoption of MPS in scientific research. And our collaboration across industries makes us a powerful force to forward the 3Rs. So this is just an example of some of our board of directors. And our Microphysiological Systems initiative seeks to increase adoption and regulatory acceptance of MPS. And this initiative includes greater than 45 member institutions, some of which you can see here, composed of over 70 members. And our initiative aims to provide thought leadership and expert consensus, facilitate appropriate discussion, collaboration, and sharing, create direct engagement between developers and regulatory agencies, develop external partnerships and collaborations, and create resources to facilitate increased engagement. And so we accomplish this via our 3 work streams. And I'll just be quickly going over some of what the members from these 3 work streams have accomplished over the past couple years. So first, our end user engagement group. So starting in 2021, we established this partnership with the IQMPS and hosted our first workshop on liver MPS. We hosted the reception at the MPS World Summit, hosted 2 webinars and 3 workshops with the IQMPS. And then in 2023, we conducted a talk at the MPS World Summit, as well as have since conducted quarterly workshops on the blood-brain barrier, vasculature, GI, and lung. And you can actually check out our presentations page to see recordings of these previous workshops. Our regulatory group has presented at the MPS World Summit on a roadmap to regulatory acceptance and began a publication on this, continued writing publications, and engaged with various regulatory groups, including the FDA, NIST, and CAT, and presented at the 3D Tissue Summit. And in 2023, continued with this publication focus on regulatory acceptance, moved toward establishing a PPP, and spoke at the 3D Tissue Summit on regulatory panel. And finally, our education group has presented at the MPS World Summit, continuously created MPS LinkedIn posts for education. In 2022, we represented the USA at the Regional Update Panel on the MPS World Summit, continued this education with webinar series, and as well in 2023 recently published a 1-pager on the FDA Modernization Act and at the World Congress. And a key part of this is actually our tech hub, which I'll paste the link in the chat soon. And so quickly, just before I hand it over to David, if you do have questions, you should be able to see the Q&A button at the top of Teams. If time permits, questions will be welcome at the end of each speaker, but if you don't get a chance to ask a question right after, the Q&A button is where you should go. And this is the last webinar, the lung one for 2023, but we will resume our webinars in 2024. And feel free to email me. My email is here, and I'll also put it in the chat if you're interested in joining our MPS initiative or becoming one of our sponsors.
Great. Okay. Hi, everyone. My name is David Kukla. and I am part of the IQMPS affiliate, and I specifically serve on one of our work streams, the Strategic Partnerships and Communication team. So I just wanted to give a quick overview of the IQ. So the IQMPS is currently composed of over 25 member companies, and we have diverse representation from drug safety, 3Rs, ADME, as well as PDPK. And here I have listed our 2023 leadership, but besides our leadership team, we also have a steering committee, and that is made up of 2 individuals from each of our member companies. And then to the right, I also just highlight a little bit more about the IQMPS. We have over 115 active member participants from the different member companies, and they meet several times throughout the year. Additionally, the IQ has 3 publications and currently actively working on several more. Also, we recently created a LinkedIn page, so feel free to follow us on LinkedIn to keep up with some of the activities and news from the IQ. So the IQ has 4 main goals. One is to serve as a thought leader for both MPS developers and stakeholder organizations. Also to provide a venue for appropriate cross-pharma collaboration and data sharing to facilitate industry implementation and qualification of MPS models. Also to create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS, as well as develop external partnerships and collaborations to help enhance the inclusion of industry priorities. So the IQMPS is made up of 5 work streams, including manuscript publications, regulatory outreach, proof of concept studies through pilot project teams, a strategic partnerships and communication team, as well as a landscape gap survey team. So here's an overview of my work stream, external partnerships, over the past few years, starting back in 2018. But however, as I mentioned, we have several other work streams and activities within cross-pharma collaboration and data sharing, regulatory agency engagement, as well as thought leadership. But then some of the highlights from our external partnerships team, in 2021, we co-partnered with the 3Rs Collaborative and established an MOU, which allows us to work together on webinars and workshops such as this. In 2022, with the 3Rs Collaborative, we had 4 webinars on different MPS systems, such as neuro and liver. And then within this year, we also had 4 workshops with blood-brain barrier, vascular, and GI, including lung today. And then additionally, last year, we invited stakeholders to participate in an IQ-MPS FDA workshop. And during this workshop, we discussed things such as where and when animal-based MPS may benefit from the advancement of the field and the establishment of confidence in the predictive nature of such platforms. Also, we compared the perspectives of the IQ-MPS and FDA on animal MPS, as well as provided examples where in vitro models are aiding in 3Rs efforts. So we're also really excited to continue these efforts throughout the rest of the year and upcoming years. So if you have any questions specifically on the IQ-MPS, please feel free to reach out to me, or you can check out our website that I have listed below in the lower left corner. Thank you so much.
Perfect. Well, thank you very much for having the opportunity to explain what I do at within my consulting business, which is Roper Toxicology Consulting Limited. I'm based in Edinburgh, UK, and I'm going to talk a little bit about what I do with respiratory toxicology. Just a bit about my professional experience. I'm one of the founding members of the North American 3Rs Collaborative, which, as you know, is now the 3Rs Collaborative. I'm one of the board members of the 3Rs Collaborative and also on the NC3Rs board. So it's good to be able to keep these 2 organisations close together. I'm a vice president and board member of the European Society of Toxicology in Vitro, and I'm a European registered toxicologist. a fellow of the Royal Society of Biology, and a Society of Toxicology and ACT member. And if anyone's at ACT next month, it'd be great to catch up with you in sunny Florida. I'm also in the Roundtable of Toxicology Consultants, and I peer review lots and lots of different papers across in vitro toxicology space. And most recently, I've become the chairperson of Peptimatrix, and I'm just going to mention something about that, which will hopefully help you with your generating innovative solutions. So what do I do, though? I provide expert support and advice to industry and agencies. That starts with 3 R's: considerations and strategy. A lot of that really is around 2 areas, and that's replacement, but also in integrated toxicology. So where we're using the strengths of the animal and the strengths of the human in vitro together. My areas of expertise are dermal absorption and toxicology, general and acute chronic toxicology, and of course, as we're here, respiratory toxicology and organs on a chip. I evaluate companies and products and services, and I'm also an expert witness performing defence for big industry. My customers come across from right across industry boards. They include multinational and mid-tier pharma, biotechnology, cosmetics, crop protection companies, household and consumer products, CROs, and a lot of you guys know me well through my support of your work through either directly with you or also through the 3Rs collaborative. I also do quite a bit of work with regulatory agencies as well. And this is usually really to help them to understand about these new technologies and how we can gain regulatory acceptance of them. What do we need to do together to gain that confidence? Also work with venture capitals. Usually I'm looking at businesses for them to acquire or to build on their current business flow with different companies, trying to get them to mix and match. And also in a competitive landscape. I'm working quite a bit with academia now, but they tend to be where they're thinking about spin-outs, and Peptimatrix is a good example there. And I'm not going to let you read all these. You need to go to my website, but there's some kind words that my customers have said, and as you can see, they're across from pharma and academia and CROs there. So the projects I work on are diverse and complex, and when I started my business, I really thought I couldn't do that many things, but you soon find out that you've been evaluating companies and products and services for over 5 years. So and in this area we have well over 100 companies in the on a chip space, so they need to be kept to look and see how they're doing, and also to try and understand where they're going to develop in the future. We're not going to stay in this fragmented space, I'm sure. I evaluate competitive landscape, find technology providers, help understand their technologies, and then report this to the client. Develop testing strategies or new test methods, and of course I do some project managing. I've been writing quite a lot of peer review and manuscripts recently, but also help on improving reports and manuscripts too, and support those regulatory submissions. Ultimately, if we don't take our submissions to the agencies, they aren't going to know what we can actually do here. And it's really important that we stand up and say, look, we're not just doing these for our internal purposes. We want these to be taken to regulatory agencies. I do a lot in quality and process improvements. This is especially important in small businesses, especially spin-outs where they haven't ever had this process before. And it really helps with giving confidence to their new customers. And indeed, I've created quite a few continuous improvement programs over the past few years, and I mentor and coach. So these are the sort of examples of things that I do. We know this conceptual model of how we do a risk assessment with a rodent. We expose them to. I'm just going to change my. No, it's not going to work. Try and change my. No, I'm going to give up. Right. So I don't know if you can see my pointer here, but we dose an animal, we do a human equivalent concentration and make a risk characterization. Now we actually have got a new way of doing this. We don't need to use the rodent. We can use we can get all the information together and do this through a much more complex, much more human relevant system. So we can look at particle distribution of inhalables. We can do the inhalation exposure into, in this case, it's a 3D model mucilaire with our in vitro test, and we can do lots of different endpoints. CFD modeling, computational fluid dynamics. If you're an engineer, you'll be bored of this. If you're not, you're a biologist, you're dead excited. Human equivalent concentration and characterization. And here's a great example of where we've taken this through. This was for chlorothalonil with Syngenta, and we've now got this as a case study with the OECD. And this did actually result in a full replacement of a 90-day rodent inhalation toxicology test, and it was far more human relevant. So that do have a look there. What we're particularly happy about is that we've got a section in there about considerations for using respiratory toxicology NAMs. Very important papers to look for. This one here with Amy Clippinger and a lot of people across industry that you may or may not be aware of, but we're really pleased it covers so many different areas of thought: target site exposure, molecular initiating events, etc., etc. And we've got also a nice little decision tree for you to follow. Some of the work when I was at Charles River, we've just published only recently. And you can see here that you can see the effect with TIR on the concentration of SDS, and it remains over the full week. So although there is some recovery, we can see the same thing happening in LDH release. And pathology is especially important because we want to relate our in vitro models in with our in vivo models, where we're really used to making those decisions on pathology. But why do we need to actually just do respiratory? Well, this paper here was we actually did multiple things. We looked at skin corrosion, irritation, eye damage and irritation, skin sensitization, as well as looking at the acute airway. And we were able to again use this with a rat in vitro model, epi airway. We were able to calculate the same things, benchmark dose levels in order to calculate human safety. More recently, we've just published work where we're demonstrating the confidence that we actually have in our in vitro models. So this compares IC50 to IC25s of 14 different chemicals with MatTek and Charles River. They stick together nicely, and the R-squareds are very good. So we've got this confidence that we have good interlab there's low interlab variability, and we swap this around, and you can look at the effects of the human versus rat. And again, we've got good variability. We were actually able to predict the 14 chemicals very similarly with the human and the rat models. So we're very pleased that we got a nice tox spotlight there. Just a couple of weeks ago, I published an opinion paper with my colleague Dan Neal in Dundee, and we're looking for the potential to use sputum mimics to allow us to start to look into improving our ability to perform chronic and repeat dose testing. If you're very interested, it'd be great to talk, because we're looking for some ideas and how to get some funding on this. We think that acute's a long way forward, but we now need to do something in chronic. Taking a bit of a step back, we had a workshop a couple of years ago, and we've just published it. I think it was just this year, and it's incorporating new approach methodologies into pharmaceutical safety testing. The group that I was in was in the respiratory team, and we've identified different processes, exposures, different diseases, and we've able to look at the effects, how to measure them, what our cell sources, our analytical platforms, etc., are all in here. And these are already commercially available tests, so you can do a huge amount of information in vitro with human tissues, before you even start going into man, you can actually generate huge amounts. And this is where we're starting to talk about human as the second species to replace maybe the non-rodent species. There is also in their cardiovascular, central nervous system and liver. So have a look at that. Developing new tests and an animal-free chemically defined media. How can we create new test methods when there are so many animal-derived media? And that includes things like matrigel. And if you don't know what it is a mouse tumor, and fetal bovine serum albumin. And again, that is a Schedule 1 process in Europe. If the best place to go if you want to know about this is to go to the NC3R's website. Just type in, I think it's scaffold, and you'll get this space. And you can see there are lots of different companies involved in animal-free, chemically defined. And I'm just going to focus on Peptimatrix because I'm now the chairperson there. And we've just launched our first new media, which is to directly replace matrigel. We just launched it last week. And I've just been told we've got our first sale, and we've also got some projects starting in the respiratory space. So really be great to talk to you. You can easily reach out to me. As Laura just said there before, join the 3Rs collaborative. But if you're not going to, you can still learn a lot from us. You can go back to all those historical webinars and workshops that we've been performing. But do reach out, do come and join us. It's a really exciting place to be. You'll meet lots of really interesting people, very, very focused on all levels of 3Rs. Or if you're specializing in a space of the organs on a chip, then come and join us. And if there are any questions, I'd be delighted to answer. Otherwise, we're 2 minutes ahead, and I'll be. We can always talk.
Yeah, it's a pleasure to be here this afternoon. Well, actually, morning for most of you in the North America. So yeah, I'm really happy to have the opportunity to talk about some of the work we're doing here at ImmuONE, specifically around animal-free tools for inhalation risk assessment, with our overarching aim to make every breath safer. And my name's Lewis Scott, and I look forward to sharing some of this work with you. So we focus on alveolar macrophage responses in predicting inhaled safety. And safety with regards to what is assessed in the lungs has typically been assessed using 2D epithelial cell culture. And whilst these are convenient models, they really have limited endpoints, typically using tear or cytotoxicity. And the issues with these are also that they're not sensitive enough to determine the adverse effects on lung health, especially over long-term administration or changes in inflammation that happen during that time. So that leads us to the influence of the alveolar macrophage. And the alveolar macrophages have really key roles in everything from particle clearance to the homeostasis and resolution of inflammation within the alveolus. And because of their role, when we're starting to include them in our assays, we're able to identify chemicals that could be harmful, but not necessarily causing obvious toxicity as detected by the 2D epithelial cell culture. So that brings us to the work we do here at ImmuONE. And we do a number of things. We have a number of arms. And our first arm, if you will, is our immune-focused cell culture models, which we are currently in the early stages of development and hoping to have them commercially available. And I'll talk a little bit about them in a minute. We also have our tailored assessment strategies. So we work as a contract research organization as well, and we solve various problems, weird and wonderful, that come our way, everything from the mouth and the nose all the way down to the deep lung, with a focus on solving all of those problems and providing consultancy in that space. Then we have our novel assessment approaches, so our multi-endpoint analysis. And that's specifically what I'm going to talk about today. Our high content image analysis is a tool for categorizing compounds as either safe or unsafe earlier in a drug discovery pipeline, so that we can minimize the use of animals in vivo experiments. So our models, we we'll spend a little bit of time on this, but I'm only going to focus really on two of them because those are the ones that we have ready, which are ImmuPhage and our ImmuLung. So we build our models up layer by layer, creating a platform of models with increasing complexity. But as you get more complex, so the cost increases and also some of the technical difficulties of working with them. So we start off with our alveolar macrophages in monoculture. We build that and we call that our ImmuPhage. We build that up to make an ImmuLung. Remember these names, I'll be testing you later. So our ImmuLung was our alveolar macrophages with the epithelial cells in co-culture. And then we're currently in an R&D stage of making an ImmuLung FT, which is our alveolar macrophages with the epithelial cells and fibroblasts, or the immufibrosis, which is that ImmuLung FT model in an inflamed state to model fibrosis. But specifically today, we're going to focus on the ImmuPhage and the ImmuLung. So the ImmuPhage was spawned out of limited immune models for in vitro lung safety assessment. And we wanted to create a model that would allow the alveolar macrophage to be assessed in terms of a tool for safety assessment. So features of the ImmuPhage is terminally differentiated, and you can see this graph here showing the amount of viable cells versus the time and days, and it's terminally differentiated, unlike monocytes or maybe some existing protocols in this space. It has some phenotypic markers of tissue resident macrophages. It has a high phagocytic function and a standard response to challenge. So if you stick something like amiodarone on it, it will upregulate the phospholipid content up to 5 weeks in culture. Or if you stick something nasty like staurosporine on there, then it's going to still be cytotoxic 35 days later. And then the ImmuLung model, just a little bit of data about that. So it's an alveolar epithelial barrier with immune competence. So we have that barrier function that's maintained both at the air-liquid or liquid-liquid interface. And here are some of the fairly high tier values maintained up to the 22 or so days in culture, both at the liquid-liquid and air-liquid interface. It maintains functional tight junctions, so low apparent permeability. But then when you treat with something like EDTA, that's going to increase the permeability. It is immunocompetent, so there's cytokine presence in there, both, I guess, constitutively at a low level. And if you whack a TLR4 agonist LPS on there, then you're going to have upregulated levels of TNF alpha, IL-8, IL-1 beta, and IL-6. And then finally, various stimuli. If you chuck all sorts of stimuli on there, amiodarone, staurosporine, there's going to be various changes that we can detect in the ImmuLung. So for example, staurosporine increasing cell death and also increasing the amount of apoptotic cells that we can find. So those are the two models that we're going to be using today, and we're going to talk about as MPS approaches. So the problem that was faced was the requirement for a tool for safety profiling, and the problem centered around the foamy alveolar macrophages that were being constantly observed in preclinical rat studies, and actually were causing a large number of compounds to actually halt their progression into the later stages of development. I was unsure whether these were adverse or adaptive responses. And we wanted to generate a tool that was going to be able to identify compounds as safe versus unsafe and identify foamy macrophages earlier rather than having to proceed into in vivo studies. So our solution was using a cell painting approach to identify foamy macrophage phenotypes. So the protocol in brief, basically you take some alveolar macrophages, you expose them to your compounds of interest, stain them with a cocktail of fluorescent dyes, and undertake some high content image analysis, which can give you epithelial dependent or independent morphological features of your alveolar macrophages. And there's a little paper down here, which I can share at the end, and you can all read if you so wish. Now, the power of the high content image analysis is combining several features to generate a fingerprint. We're not limited by just having one individual descriptor. We use 4 typically with our Morph1 assay, which are mitochondrial activity, vacuolation profiles, and cell area. And that gives us detailed understanding of cells within the population rather than just the averages of a population. Now, this graph here is a fairly representative heat map of the phenotypes that one would observe in macrophages in our immuphage versus our macrophages and epithelial co-culture, and we can see that there are no real discernible differences between the two experiments there. So what we find is that if we were to add something like cigarette smoke on there, then there's a very noticeable visual change in that fingerprint, in that barcode, and that's correlating to changes in individual cell populations, specifically with regard to changes in mitochondrial activity and their vacuolation profiles. So we'll look at that data in a little bit more detail. So first up, we have our control. Data from that, and what we can see is when we look at the phenotypes in a bit more detail. Now, these are phenotypes that make up more than 1% of the total population. We can see little changes between the macrophages on their own, the immuphage, and the ImmuLung, so the macrophages and epithelial cells. So not really many changes in these profiles in these populations. Yet when we add something like cigarette smoke, then there are quickly some very discernible changes. So there's a general shift from the right to left, which correlates to the lower mitochondrial activity. Now, we'll zoom in on some of interest. So, for example, this one here, which profiles correlate with decreased mitochondrial activity or decreased mitochondrial activity with increased area of vacuolation. Some other ones of interest here are the epithelial cell dependent decreases in mitochondrial activity, but also increases in cell area, vacuolation, and the total number of vacuoles. So the general decreased mitochondrial activity and increased cell area, we're indicating that's epithelial dependent autophagy, as there's mechanisms in the literature that suggest that autophagy can be affected in alveolar macrophages when exposed to cigarette smoke. Now, we use this approach to benchmark safety and the safety of our immuphage responses to inhaled compounds, because our immuphage will exhibit different responses to inhaled drugs and cigarette smokes and various compounds known to have adverse effects in vivo. And we can use this to differentiate cellular responses, and we can phenotype them and elucidate the toxicity mechanisms. And we can see that there are noticeable changes between things that are known safe and things that are known unsafe. And that gives us a tool to predict safe versus unsafe responses. Now, the final thing I'm going to talk about today is a paper with the overarching aim of using cell painting as a tool for categorizing compounds as either safe or unsafe for safety assessment and facilitating go to no-go decisions before any costly and unneeded in vivo studies. So what we did is we looked at morphological assessments of alveolar macrophages and also lipid assessment, intracellular lipid profiles of our alveolar macrophages to see if we could identify foamy macrophages before going into in vivo studies. So we took some generally safe compounds, should we say, beta agonists and muscarinic receptor antagonists we use, two classes of drugs which are used to treat long-term respiratory conditions. There are negative controls, as they are usually without any adverse alveolar macrophage responses. And what we found is there's no major changes to either the vacuolation profile or some of the functional responses such as phospholipidosis or mitochondrial activity changes with these drugs. We then took some known unsafe drugs. So in this instance, we took the cationic amphiphilic drugs, or CADs, which are phospholipidosis inducers. Now, these aren't inhaled therapies, luckily, but they are model drugs for phospholipidosis. And amiodarone specifically can cause fibrosis and various responses in the clinic. So we were able to find that this tool was able to predict the increased phospholipidosis in these cells, and actually was used and could be able to be used before actually having to go into any in vivo studies. So the idea of this was born out of needing a better decision-making tool to reduce the number of non-developable drugs entering that expensive testing process, and will allow us for better decision-making in the early stages. So less unsafe compounds going through, and ultimately less animals going through. So yeah, please get in touch with me if you'd like to learn any more about all of this. I appreciate there's a lot of info there. Our website is at immuone.com, and thank you so much for NAC3Rs for having me present today.
Great. Thanks so much, Lewis. And we do have a couple minutes, and there's a few questions in the chat. So feel free to keep your camera on, and I'll just read the first one out loud to you. How is the TER measured across an air-liquid interface? Was it by adding a liquid, media PBS in apical chamber for a short duration while taking the measurement? Just let me know if you could.
Yeah, no, that's a fantastic question, Raggi. I will yeah, I'll have to get back to you on that one after further consulting with the scientists on that. I'm not actually in the lab at the moment, so I'm not sure the technical intricacies of that, but I can definitely respond to that question later.
Great. So something maybe to follow up on.
Yeah.
And we have another question in the chat. Observations of foamy macrophages are not considered adverse by FDA as an in vivo endpoint. Is this a correct endpoint for MPS systems?
Yes, well, it's not necessarily adverse. This could be an adaptive response on its own, or even having this as a manner of identifying these early on, so that we can have some mechanistic understanding of, okay, is this compound going to elect upregulated phospholipidosis? Is it going to cause changes? Is there going to be some alterations to foamy to macrophage phenotypes? Is going to be a very, very good tool to incorporate into the MPS system, so that we can understand more about the drug before it goes into the later stages. I hope that answers that one. Okay.
Thanks. And maybe we can quickly squeeze in the last question. For your safe and unsafe drugs, did you match exposure levels lung in the in vitro models?
Yeah, a great question as well. And yeah, this would also be something, Mary, that I can follow up with you on later on, to see about the exposure levels and if they were matched between what's been found in the lung to the in vitro models as well. And I will, yeah, certainly follow up with you on that one.
Perfect. All right, so hello everyone, my name is Giulia. I'm from Alveolix, and I'm happy to be here today to give you a brief overview of our lung-on-chip model and its applications. So first, very briefly, a short introduction about who we are. We are a startup, a biotech startup developing organs on chip. We are based in Switzerland, where we have 2 locations: in Bern, the capital, where we have our biology and research laboratories, and in Biel, where we have our clean room and production site. We provide organs on chip technology to mimic any organ barrier, basically. So our focus is support and forces to create the motion, especially for then creating the best in vivo-like environment as close as possible to the human body. So we focus really on this motion and forces and support and microenvironment to have better models for preclinical studies. So our technology we developed is called a barrier on chip system, and it's made of these components that you see here in the picture. There is an actuator and an exchanger. These 2 are electropneumatic units that are the controlling units for the motion. So the actuator controls the motion, so stretching and so on, while the exchanger is more for controlling, for example, medium exchange, measurement of the TER, and so on. These 2 controlling units are connected to the chip per se via this black support that you see, which is we call dock. It's a docking station, basically. And your chip, where with the cell culture and everything, will be placed inside here. And I will explain it to you in 1 second. So the way we developed our technology is in a way that you're free to set your parameters for the motion, so for the actuator to create the motion that you want, and is more accurate for your models. So you can have, for example, uni or bidirectional membrane movement. You can have different stretch levels for mimicking a normal, healthy system, or more towards a pathophysiological stretch level. You can also adjust the frequency and also have active versus dormant time periods of stretching. So all of these parameters are easily customizable for every different application, and that's why with this we can mimic different organs of the human body. So here, just a brief introduction about the chip per se. So this is the consumable. It's called IX12 because it's made of 12 independent units. And here you see what we call a unit. So one unit is made of inlet and outlet wells, which are mainly used for medium exchange and also sampling of supernatant for their analysis. And in the center, you have a cell well, which is divided in the middle by a porous membrane. So this creates an apical side and a basolateral chamber. And this membrane is very thin and it's porous. And since it's just 3.5 micrometer thick, when you culture, for example, epithelial and endothelial cells on the two sides of that membrane, you can create very thin co-cultures, so double layers of cells. And since it's so thin, the cells can are really in close contact to each other. So that's one of our features. So here is just a video to show how the stretch movement is created. So this technology that we created was inspired by the human breathing, actually. So basically at the bottom of the basal chamber of the chip, there is a cavity with a membrane on top. And this membrane is deflected into the cavity, controlled by the actuator, which follows your setting that you set. And this movement, since during breathing the chamber is closed, this movement is then transmitted to the blue membrane here in the schematic, where the cells are cultured. So that's how then the cells get stretched. And you see here on the right a movie showing these were lung epithelial cells under stretch. So the settings were set to the standard human normal breathing frequency and strain. And of course, as I said before, you are free to change the settings and parameters to then mimic different frequencies and stretch levels for other organs. Our chip is actually very easily dismounted and disassembled in different parts, and this can this allows also high resolution imaging, for example. Also, the membrane can be embedded, and then readouts like immunohistochemistry can be done on the cells. We can also do, of course, all the live cell assays, like toxicity, viability assays. We can sample from the chip, both compartments, so then we can check for cytokine release, for example. Of course, we have also barrier functionality measurements like tear and permeability assays. And we also recently developed a protocol for detaching the cells from the chip and then still have them viable and available for further single cell sequencing or bulk cell sequencing, or even for flow cytometry analysis. So also what I didn't mention so far, the chip is 96 wide plate format, so it's also fully compatible with any microplate reader for also live measurements or live imaging. So here now, if I go more in the lung chip models that we developed, actually we started with the lung as our first organ when we started with the company. So we have a lot of expertise on these models. In this time, we've developed some models based on primary tissue, so primary alveolar epithelial cells, for example, or primary endothelial cells or fibroblasts. Else, we also have models based on cell lines. So our main focus is normally, and expertise also, is about alveolar models. However, we also establish some models for small airways, for example. And regarding alveolar epithelial cells, we developed also a cell line derived from primary cells, which has the characteristic of type 1 and type 2 cells, alveolar cells, and also provides a stable barrier over time. And this is also available as a product for our partners. We call it cells on chip. So the partner gets the chips already seeded with the cells ready to use in their lab. So in both these cases, primary cells or cell line-based models, we can make it more complex by having co-cultures with endothelial cells or fibroblasts, for example. And we can also add immune cells to the system, depending on the application. And we mostly use for these PUMCs from the blood or THP1. So here I will show you briefly some applications in different areas that we already did on chip. So for example, one of them is about safety assessment of immunotherapeutics. And here we used one of our immunocompetent models, so based on alveolar primary cells. And we tested some immunotherapeutics such as TCBs, so T-cell bispecific antibodies, or ADCs, antibody drug conjugates. And here I have some data as example with in the TCB studies, safety assessment studies, where we found that the TCB tested was causing barrier breakdown, cytotoxicity, also cytokine storm, and also what I show you here in detail is that we were able also to count how many immune cells were recruited specifically at the epithelial layer. And also among these ones, we were able to harvest them and run a flow cytometry and then analysis, and then we could check for activation. Another example of an application is in the field of pulmonary fibrosis and interstitial lung diseases. So here we use, for example, TGF-beta-1 for as a trigger for building up the disease status, and then this model can be used for drug efficacy testing. Some data about this, for example, here we see that upon addition of TGF-beta, the barrier gets damaged. And we compared in this case non-breathing and breathing conditions, and we could see that in breathing conditions, the barrier damage was visible earlier in time and also more pronounced in terms of in extent. So upon TGF-beta treatment, we also saw pro-collagen 1 and collagen 1 secretion increased. And as proof of concept, we then treated with nintedanib, an approved drug for the for fibrosis, and we were able to show the reverted trend in secretion of the ECM proteins. Another application and also different way of administration of drugs is inhalation. And this can also be used for disease modeling, for example, for smoke-induced COPD, or also for safety assessment of new chemicals or new compounds, for example, that are inhaled. And for this, we developed together with Vitrocell a cloud X12 system, which enables nebulization of compounds directly on the cells on the chips. And here an example is with PHMG, which is a toxic chemical that was used as disinfectant for air humidifiers, and then was found to be toxic. And here we were able to reproduce a clear increase in cytotoxicity, and also gene expression, inflammatory genes were overexpressed. We then treated with fluticasone 24 hours after, also by nebulization, and we could show that we were able to revert the trend in cytotoxicity and also in the inflammatory genes. Another application that is possible to do on the chip is infection. So we have done both bacterial and viral infection on our chip. Now, today I show you just some data about SARS-CoV-2 infection. So here this model was done on the cell line on chip. alveolar cell line, and it was done also in air liquid interface and static versus dynamic. So here the infection efficacy was compared, and actually in dynamic conditions it was more, the virus was produced more. And also when checking what happens to the barrier during the infection, 48 hours after infection, the barrier was impaired in static, but more pronounced, the drop was more pronounced in dynamic. And also, again, as proof of concept, we treated with remdesivir, which is well known for COVID treatment. And here also the trend, the barrier damage was reverted. So here I just have a few minutes to show you just some case studies to show you the impact that can have this technology on industry. So we had a case, for example, where our customer came to us with a drug that was already in late preclinical phase, but then was stopped because it was showed to have severe side effects on non-human primates, but no other preclinical model was able to predict that before. So we tried that on the chip, and we were happy to see that the chip was able actually to also predict that toxicity. And actually, these data were already used for 5 IND submissions. Another example of a case study is when customers have contradictory results in preclinical animal studies, and the chip can help them in the decision making. So it can help to decide how to go on with the drug, being a more human in vitro predictive model. And a third example here is there was a drug. The customer had a drug that was showing some side effects in the lungs, but only in a fraction of patients. So not all of the patients were answering the same way. And we tried this drug on chip on different donors using the primary cells models, and we were able to reproduce this donor-to-donor variability on the chip, showing that also these kind of studies are possible with our technology. So that's it from my side. Thank you for your attention, and I'm happy to get any question per mail or later in the discussion, since I think I use that all the time.
Thank you very much to the organisers. It's great to see all the fantastic work that's going on in the lung in vitro space. So I am presenting the PhysioMimics models from SynVivo. So anyone who doesn't know about SynVivo, we're an organ-on-chip developer based in Cambridge in the UK. We've been developing these models for a while now, with many world-leading collaborators to make the PhysioMimics system, which is our platform. And our main focus is to provide solutions for the challenges that drug developers and researchers have in the field. And the main applications we focus around ADME, safety toxicology, and disease modeling, with our three current main organ models being the liver, the gut, and the lung. Although, as I said, we're an R&D company, so we're constantly developing these models as well. The PhysioMimics system looks a bit like this. For those of you who haven't seen it before, let me just get my pointer. So we have a controller, which is this PC-looking box. This sits outside of the incubator, and this is where you control your pneumatics. This is translated into docking stations, which sit inside of the incubator, so you can have 2 of these per system. And within these docking stations, you can fit 3 drivers, which house our consumable plates. This is an example of one of our plates, which I'll go through in a minute. This is a 12-well individual 12-well chip plate. So within each of these wells or chips, you can house your different tissue models. So in terms of the organ models, I'll skip over this because obviously I want to concentrate on the lung, but our liver model looks slightly different, whereby you have perfusion running up and through the liver microtissue, and you form these beautiful donut-shaped liver microtissues. Our barrier model plate, which houses our lung and also gut systems, are an insert-based plate, so you can use 24 well size things like trans wells or other commercially available inserts. So these sit within each of the wells, and you have your perfusion going round and underneath each of the inserts. And as I said, these are all individual wells, so all have their individual micropumps. That's a nice example of the gut. We also have a dual organ model, which you can see here. This is a 6-well plate where you combine the technology of the barrier and the liver plates together. So there are 3 micropumps, one under the barrier side, one on the liver side, and then one which connects the 2 together. And these can all be turned off and on individually and independently. So you can do some quite powerful studies using these, which I'll touch on later in the presentation. So back to the focus, which is our lung MPS. So we currently have 2 main models within the lung, which is our bronchial airway and our alveoli. These being 2 very distinctly different areas of the lung where we see requirements for modeling. And we use primary cells within our system, commercially available primary cells. And when we first developed these models, we used these cells in both a static and an MPS condition and cultured the cells for 14 days at an air liquid interface using the same media. And here you can see that within the static, we don't see great tissue being formed over 14 days, although we do know that later on, as has been shown by other providers, sort of 4 to 6 weeks, you get good differentiation in the bronchial model. When we put these cells into the MPS over the same 14-day period, you get this greatly enhanced differentiation to a pseudostratified epithelium in the bronchial model. And then in the alveolar model, these beautiful alveolar sac-like structures, which are formed over the 14-day period. So to go into a bit more detail, our bronchial MPS, we've sort of built all of these models up from a sort of basic to complex. So here you can see just the bronchial epithelium where we culture our bronchial epithelial cells on the apical side of the insert at air liquid interface. And you can see over 14 days, we get differentiation of ciliated cells, which have beautiful ciliated structures, goblet cells, which produce mucus, and other cells such as club cells and basal cells. And together they form these beautiful pseudostratified epithelium, which are highly functional, producing, as I said, that mucociliary barrier, which is really important for looking at ADME, but also disease modeling as well. And then our alveolar model, which again, this is just the epithelium at this point, again, just within the apical side of the insert, cultured over 14 days at air liquid interface. So when we developed this model, we were really interested in making sure we had the right cell populations within the alveolar model. So particularly focusing on the differentiation to type 1 and type 2 cells, with type 2 cells being a sort of key subtype, because often we find that in other models, these cells quickly transdifferentiate into type 1 cells, so you really get a lack of that population. But within our model, we're able to keep this population. You can see a really good population of them, and these remain stable over time, producing things like surfactant. And obviously, these cells are also really important for the inflammatory response as well. So then we can increase the complexity, and we have included the endothelium on the basolateral side of the insert. And what we find is that inclusion of this endothelium really enhances the model in terms of the physiological relevance. So within the bronchial model, you can see it enhances the tear quite drastically. You still get really good differentiation of the epithelial tissue into the different subtypes, you can see here. And you can see in this imaging here, hopefully the video is working for everyone, but you get a really nice layer of those endothelial cells on the basolateral side, which you can also see in the alveolar model here. They form these really flat but continuous structures along the basolateral side. And then to further increase complexity, depending on the research question, we can also start to think about an immune axis. And so because we have this circulating flow, we can add in circulating immune cells to act like peripheral blood-like cells, such as PBMCs, circulating monocytes, etc. And what we've also done in the past is to add monocytes into the apical side of the alveolar epithelium to act like alveolar macrophage-like cells, which you can see here in this imaging in the magenta color. And these patrol around the top of the alveolar epithelium, as we see in humans. And we've looked at the different responses of these models, sort of with and without monocytes. So adding classical TLR agonists such as poly-IC and LPS. And what you can see is that the two models react very differently, as we'd expect, in terms of their inflammatory response. Something key to point out within the bronchial model, which was great to see, was that when we applied LPS to the bronchial epithelium, we didn't see any inflammatory response, which is what we expect because the bronchial epithelium doesn't express a TLR4 receptor. However, when we add in our monocytes, we do see that response occurring, and we see that crosstalk between the two populations. There we go. So a key part of what we do at SynVivo is really making sure that the models we make are really robust and well validated. So we've looked at our models over 28 days. So this is an example of our alveolar tissue. And you can see as an example that our tear is extremely steady over those 28 days that we tested them for, whereas the static model is sort of going up and down and then eventually crashing after about 20 days. And another thing that we do in-house to really sort of assist our customers with their use of MPS is to validate those primary cell donors of our epithelium and our endothelium. So again, here you can see an example of our alveolar set of donors that we tested in-house. Is an example just as the IHC images that showed that this donor 2 did not pass our criteria, and we also saw this within the cell populations that we tested within this qualification. So in terms of applications, currently we have a disease modeling application looking at infection and inflammation. So the main sort of point of this is to look at how the different areas of the lung are infected, how it might affect the tissue health over time. And we can look at acute and chronic infections over time and really pick out those points at which different mechanistics happen. And then in terms of our other application, which is ADME, looking at inhaled medications, the main application here is to look at how drugs accumulate or permeabilize through the lung epithelium and endothelium. So here you can see an example of a short-acting beta-adrenoreceptor agonist after 24 hours, where it was mainly found in the media, where it had basically permeabilized through. And then a long-acting corticosteroid, which was mainly found accumulated within the epithelium, as we'd expect it to, because it enacts that long-lasting effect. And the joy of our system is that it is open well and it is an insert-based system. So you can do things like aerosolized drugs or pathogens onto the system, or you can simply add a liquid dose to the apical side. And it's not enough that we qualify these assays. We actually want people externally to qualify these as well. So we have long-standing collaborations with different partners. So our disease modelling side, we've partnered with ICON at the Liverpool School of Tropical Medicine, who are currently using our system for looking at different COVID variants and being able to describe the characteristics of these variants using the system, and then looking at drugs and the efficacy of drugs on those variants. We also have a long-standing collaboration with a group at the FDA CEDA, who, amongst our other models, are looking at our lung model to qualify its use in ADME. And so finally, just to finish off, to add a little bit more complexity onto what I've described, we can also do multi-organ where we add our lung into our liver system, and we can also add circulating immune cells into this to look even more depth into systemic inflammation, particularly when thinking about infections. And here's just an example data set that we did a couple of years back, looking at systemic inflammation. So we can look at things like cytokine releases over time. So this was over a 72-hour period where we opened the interconnect from the lung into the liver and allowed the crosstalk to happen between the two. And you can see these responses happening over time. We can also look at local tissue effects, whether that be the lung or the liver, looking at metabolism, tear, mucus production as examples. And then we can also quite uniquely look at systemic effects such as the acute phase response, which is obviously generated from the liver from systemic inflammation. And again, we can measure that over time and correlate it between what we see in our local and systemic populations. So with that, I'll conclude to hopefully say in this short time I've showed you the use of our lung MPS, the ability to add complexity in relation to the research question, whether that be immune or multi-organ. And the joy of the system is that it has this large sampling volume and tissue quantity, which means you get this really rich mechanistic data that can be measured over time due to the open well format of the system. And obviously there are 12 individual chips per plate, which means if you use the whole system, you've got about 72 replicates, which obviously is pretty attractive. And with that, I will finish there. Many thanks to the CM Bio R&D team, with a special mention to Hayley for her work in this area. And I'll welcome any questions if there's time, but if not, please add them to the chat. Thank you.
Thanks, Emily. We do have about 30 seconds, and there is one question in the chat. I can just ask it to you. Is there any physiochemical limitation regarding new drugs for these models?
Oof, that's a big question. Any physiochemical limitations? No, within scope. I think it's really important to understand your physiochemical properties. But the great thing about our system is it's made of COC, so it has very low binding if you're thinking about adsorption into the system. So it's particularly useful when understanding drugs with those high adsorption properties.
Maybe something that can be returned to in the roundtable when there's a bit more time to talk about this, for sure. All right, thanks so much, Emily.
Well, thank you for inviting me here today to speak. So I'm going to give an overview of the lung platform that we have at New Cells Biotech. New Cells Biotech is a CRO-based company in the north of the UK, and we work on in vitro models for drug discovery focused around the kidney, the retina, and also in the lung. So the first model I'm going to talk about is the small airway epithelial cell alley culture. I think we've kind of touched on this quite a lot today, so I will quickly go through the slides just to give you a brief overview, and then if there's any questions, please feel free to reach out separately. So similar to the models which have been presented today, our small airway alley model is derived from primary human basal cells, which are isolated from the non-cartilaginous region of the lung. So this is the region which lies proximal to the alveolar region. So we derive these basal cells from a commercial source to produce a fully differentiated in vitro epithelial model for the study of airway physiology and disease, and also for the assessment of compound toxicity. So we currently have 3 normal healthy donors, which we validated in-house. We tend to use the cells at a low passage to ensure efficient differentiation and reduce the risk of cellular senescence. The model's been well characterized and closely recapitulates the in vivo environment. It's highly reproducible. We've run the same donors multiple times now, so it allows for potential follow-on studies at later time points. We currently do a 24-well individual well format, which allows for assay flexibility. And we are actually looking now to develop this into a high throughput 96-well based culture to allow for things like high content screening. So we take the basal cells, which I'm sure you're aware are the stem cells of the airways, and then we're able to proliferate and differentiate these key epithelial cell types. And when we cut them under air-liquid interface conditions, we see that our model comprises of basal cells, club cells, goblet cells, and ciliated cells, which are known to be the key epithelial cell types of the airway regions. To do this, we go through two different expansion phases. We initially expand the basal cells in a cell culture flask, and then we seed them onto these semi-permeable supports, where we have submerged conditions. So we have media on the top and expansion media on the bottom. And then once the cells become confluent, we remove that liquid from the top to give us the air-liquid interface and begin the differentiation phase. We tend to cut the cells out for between 21 to 28 days, depending on the assay that we're interested in, and also the cell types which we're looking to study. So we can see throughout that growth period, here we've extended that out to 48 days, that we see a nice expression of key tight junctional and cellular junctional genes and proteins. So you can see here, for an example, we have ZO1, E cadherin, and JAM-A, and the expression is maintained throughout the culture period. And we can also localize ZO1 and E cadherin to those junctional barriers as well. This gives us a high level of confidence that the cells are actually polarizing correctly, and we have a nice tight epithelial barrier. And this is reflected by our tier measurements. So coming back to the question that we had earlier in the chat there, to measure these tiers of the ALI cultures, we do indeed add PBS or cell culture media into the apical chamber for a short duration of time. We then allow the cells to equilibrate to allow for any ion transport changes, and then we're able to measure the tier. And as you can see, we see this nice increase from day 0 to 7 as the cells begin to polarize and those junctions are formed, and then it's maintained throughout the remainder of the culture period. And the barrier is actually size selective. So when we apply different molecular weight fitzy-dextran molecules to the apical cell chamber, we can see that their ability to diffuse through the paracellular pathway is actually decreased as that molecular weight is increasing. We've tracked the gene expression changes throughout the entire differentiation period, and these match nicely with when we would expect to see the different epithelial cell types. So as you can see, early in the differentiation period towards the left-hand side here, we've got a nice high level of expression of our basal cell markers. And then throughout the differentiation period, these basal cells become replaced by club cells and goblet cells, with the ciliated cell markers FOXJ1 and TPP3 increasing in a linear fashion throughout the differentiation period. The cultures have nice prominent cilia, which beat nicely in culture, and we do see nice mucociliary action as well. We're able to stain for cell-specific proteins, so we can see here we've got keratin 5 as a basal cell marker, CC10, which is the common club cell marker, MUC5B and MUC5AC for the goblet cells, and we can see a nice uniform expression of acetylated tubulin across the entire permeable support, indicative of a nice ciliation of the cultures. We're able to perform toxicity-based readouts in these, so we currently have 2 internal controls. We use puromycin and adriamycin, and we're able to measure TIA, LDH release, ATP activity, and MTT. And formazan formation using the MTT assay, and assess the changes in these readouts with dose-dependent increases in compounds. And this is often something we do as a service for different clients. You can see here that we're able to add different cytokines. So this is some data that we have around TNF alpha, and this is a nice illustration of how each of our donors responds slightly differently to each treatment that we use. So we're quite selective in the donor that we would use for a study. So you can see here donor C has a much more pronounced effect in response to TNF alpha. And whilst we see this breakdown in tier, sorry, the axes are quite small there, as we see the breakdown in tier, indicative of epithelial barrier breakdown, we don't really see a significant change in LDH release, nor in ATP activity or formazan formation in the MTT assay. I think the advantages of this system is that it's very versatile, as I'm sure many people on this symposium are aware, and really it can be applied to a lot of different situations, and it kind of depends on what the study requirements are as to how we would use the system. So that's something we would work on a one-to-one basis with, depending on the client's needs and also what the outcomes of the study are. A second offering that we have here at NewCells currently is a fibroblast to myofibroblast transition assay. So we're very interested in fibrosis. So this is a high content imaging assay for the study of fibroblast activation and matrix deposition. So this is a 384 well based assay. We use normal healthy lung fibroblasts. We currently have 3 validated donors, and we're continuously validating other normal human lung donors, and we're also able to access disease fibroblasts as well. We use a disease relevant stimulus, so in this case we're actually using TGF-beta, and we include validated experimental assay controls on each plate. We're currently robustly measuring cell number. We're able to detect smooth muscle actin and also detect collagen 1. We can also look at EDU incorporation, cell senescence markers, and we can also collect the stooping agents to do biomarker analysis as well. So we seed the lung fibroblasts into 384 well plates under reduced serum conditions to control for cellular proliferation. And then we stimulate the cells with TGF-beta1 in the presence of macromolecular crowder. So the macromolecular crowder promotes the deposition of secreted collagen molecules onto the plate, which allows us then to detect them using ICC and high content imaging. So we usually pre-treat with TGF-beta, we would usually stimulate and then co-treat with TGF-beta and the test article, but the setup of the assay can be modified according to the study. And then 72 hours later, we do our readouts. So this is some of the data we have here. So you can see, we see a reduction in EDU incorporation into the cells as we reduce the serum concentration of the media from 2 to 0.5%. This gives us good confidence that the cells, any changes that we're seeing in collagen expression is actually due to treatment with compounds and with TGF-beta, as opposed to treatment in changes in culture conditions. And in the presence of our macromolecular crowder, you can see that this is reduced further, and that these changes in cell count increase in the presence of TGF-beta. We do see a nice dose-dependent increase in cell number with TGF-beta, which is indicative of lung fibrosis as well. So using the high content imaging, we're able to use ICC to detect key proteins. So we have alpha smooth muscle actin and collagen 1 here, and then we apply segmental analysis to each individual well, and we can get the readouts and analyze them accordingly. So you can see here with our TGF-beta dose, that TGF-beta1 dose dependently induces alpha smooth muscle actin expression. You can see a nice log dose response curve here, and that's reflected here in the representative images that you have at the top. And we see similar effects with collagen 1. Again, we do see donor to donor variation, which you can see represented by the 3 individual lines here. With the FMT assay, we tend to include all 3 donors in the assay, just to really recapitulate what you might see in vivo in terms of patient-to-patient variations. We're able to inhibit these responses using ALK5 inhibitors. So we have 2 common compounds, SB431-542 and SB525-334, which are always included on the plates. And when we quantify that, you can see that we see these nice decreases in total collagen 1 and also in extracellular collagen 1 as well. We've also tested the approved therapeutic compounds. So we've tested nintedanib and pirfenidone. The effects of these compounds differ from each donor, but you can see that we do see a reduction in the expression of collagen 1 in the presence of these 2 compounds, although not as extreme as what we would see with the 2 ALK5 inhibitors, which is expected given that their modes of action are off and not directly inhibiting that TGF-beta pathway. And similarly, we do see effects in the changes of alpha smooth muscle actin in the presence of those two therapeutic compounds as well. So just to summarize, this is an in vitro high throughput assay for the study of fibroblast activation and collagen expression and deposition. The standard assay would be 5 test articles. These are 7-point dose response. We can use immortalized lung fibroblast cell lines, normal healthy lung fibroblasts, and we also have disease fibroblasts as well. We're able to measure relevant markers within the total and the extracellular compartments, and we see a nice dose-dependent increase in alpha smooth muscle actin and collagen 1 with increasing concentrations of TGF-beta. And we can measure cell number and look at the assessment of proliferative capacity as well. So then moving forward, we're hoping to kind of build on these two systems to make them more complex. So we're starting to think about co-culture models with epithelial cells and with fibroblasts, and also how we can make them more relevant in terms of recapitulation of fibrotic disease as well. I'm not sure where we are on time, but that's the end of the presentation. So.
Thanks, Megan. We do have a couple minutes, about 2 minutes left. If we do have any questions for Megan, please feel free to put them in the chat at this time.
Thank you to the organizers for this amazing workshop, and it's just been a pleasure to see just some of the amazing models and the data that has been coming through. My name is Deborah Ramsey, and I work for SynVivo. I'm the Director of Scientific Development, and today I'm going to talk about our lung-on-a-chip, organ-on-a-chip model for drug safety and efficacy testing. And so as you've seen already, there's a lot of great movement in this field for applying different lung models for in vitro analyses, including things such as drug delivery and drug efficacy, but then also looking at inflammation and how immune cell responses and interactions really can contribute to how tissues are responding in effects to disease modeling and also to drugs themselves. And one of the focuses that we have with our SynALI models is looking at, both inflammation and also vascular toxicology in the lung microvasculature. And we believe that these studies are going to allow us to make, finer tuned predictions in terms of, what drugs can move forward into further preclinical model testing. So as I mentioned before, SynVivo has a wide array of different organ-on-a-chip models that include our blood-brain barrier model. We have an inflammation model that looks at immune cells and their response to pro-inflammatory mediators and to drugs that may influence inflammation. We also have a SynTumor or a tumor model that looks at both metastatic tumors as well as the role and impacts of circulating tumor cells as well as immune cells and how they're targeting different tumors. The model that I'll be focusing on today is our lung air-liquid interface model, which is shown here. The architecture, it has 3 side-by-side channels that are interconnected with a porous barrier. And we're going to utilize this particular lung model not only just for looking at some physiology and some immune interactions, but also for toxicology as well, so that we can look at the role and influence of different drugs and their responses on the lung tissue and the microvasculature. So in our small airway and our alveolar lung models, we are placing our lung epithelial cells here in the center outlet, which, as I mentioned before, is under an air-liquid interface. So we introduce humidified air within this, and then we have lung microvascular cells on either side surrounding that lung epithelium that allows crosstalk between these channels. The lung microvasculature is under a liquid interface, and so this allows us to have this lovely side-by-side architecture. When we characterize our lung epithelial models, we see formation of goblet cells, including mucus production and mucus strands across the epithelial layer. We also see the presence of cilia and cilia beat rates. And we can also evaluate the expression of tight junction markers within these as well. So we can look at ZO1 shown here. We can also look at VE cadherin if we're looking at cadherin molecules. And in our epithelial cells, we can look at ICAMs and E-cadherin molecules for the expression of tight junctions. So to develop our lung epithelial model on chip, it's a longer process. So we do start with a liquid-liquid interface, seeding first our lung epithelial cells. In this example, we have our normal human bronchial epithelial cells that are placed in that center channel. And then over time, we have the differentiation process that begins, and then air is introduced onto the tissue, and then we get full differentiation across the tissue. We can usually see differentiation beginning along the sides, and then it reaches the center so that we get a fully differentiated tissue, and then we seed our lung microvascular endothelial cells, and that gives us our co-culture model. So we have two different types of vascularized lung on a chip models. We have a small airway as well as an alveolar lung model. For our small airway lung model, it's human bronchial epithelial cells. As you can see here, they're located in the center, and this is ZO1 staining. And then we have our lung microvascular endothelial cells on either side. As you can see, they're staining for the tight junction markers, in this case, VE cadherin. And these epithelial cells can express MUC5AC, and they're also showing some cilia beats that we can see here. In our alveolar lung model, this is type 1 and type 2 human alveolar cells, again with ZO1 staining. And then again, we have our human microvascular lung endothelial cells on either side with VE cadherin staining. We get mucus production across this tissue. So with these two models, one of the things that we look at within our organ on a chip system is the presence of a fully luminized vessel. And so in these cases here, we're looking at our normal human lung bronchial epithelial cells. You can see it kind of in what looks like flat 2D planar here. But then if we do a Z stack, you can see on its side that we have a complete luminized airway epithelium that's present here. And in this side-by-side illustration, you can see the airway epithelium as a fully luminized vessel, and then on either side, the lung microvasculature also with lumens. So we have essentially 3 luminized vessels that we can then run and look at for toxicology testing, as well as for immune cells. So one of the big questions that had been asked by different collaborators had been, how efficacious were drugs on lung microvasculature in terms of ameliorating the inflammatory response? And how did that really change depending on the source of those lung microvascular endothelial cells? And we all know too that, you know, sometimes the microvasculature can behave differently than some larger endothelial vessels that, so some of the cell sources become really critical and key here, as others have mentioned. In this particular study, though, they compared a human donor source for lung microvascular endothelial cells with a murine source. And they were looking at just how these different endothelial vessels responded both by permeability through a 4 kilodalton fluorescent tracer molecule, and then also by tear. And so they would look at it whether it was with no treatment alone, or if they were going to add a pro-inflammatory cytokine, in this case TNF-alpha, or if they were incubating the microvasculature with TNF-alpha and a PKC delta inhibitor that would actually act to counteract the pro-inflammatory impacts on the microvasculature and cause you know, the barrier to not become quite as leaky. So if we're comparing the human and the murine tissues, we can see there's a slight difference when no treatment is involved. But the interesting thing is when the pro-inflammatory cytokine is added, the human cells seem to show a significant increase in terms of permeability to a fluorescent tracer compared to the murine cells. When the PKC delta inhibitor is added, the nice effect is that both of those, in terms of the permeability, are regaining their tightness. And in fact, the tightness for the murine cells are very similar to the no treatment. For the human cells is also quite similar to the no treatment controls. But when they were visualizing and looking at tear readings themselves, the interesting aspect again here is that there is a difference in terms of human and murine cells just in general for tear, but that difference in tear became even more drastic when TNF-alpha treatment was encouraged in here. And then when the PKC delta inhibitor was added, the interesting effects were that, the tear significantly increased for the human cells, indicating again a retightening of that barrier. But then the mouse and murine cells, although increased in tightness, it was not as great as what was observed with the human cells. So our organ-on-a-chip system is able to really look at some species-dependent endothelial responses in the presence of pro-inflammatory cytokines and also the drugs that may ameliorate that response. One of the other ways that we can also look at the response to lung microvasculature is what happens in the presence of migratory immune cells, and what are the species differences between, how neutrophils, for example, migrate and have rolling adhesion and migration in response to an inflammatory cytokine. So in this case here, again with our human donor, they observed that when the microvasculature was treated with TNF-alpha and was placed in response to a cytokine gradient, there was a very strong neutrophil migratory response over 1 hour, and that was very, very significant across the human cells. But the interesting factor here was that when the PKC delta inhibitor was added to again tap down that pro-inflammatory response, there were very few neutrophils that ended up migrating in response to the chemokine gradient, showing the effect that delta inhibitor had in terms of, again, ameliorating the inflammatory response. The interesting factor here, though, is when that data is compared to our murine data, the mouse vasculature definitely in the presence of TNF-alpha shows, more neutrophils that are migrating across in response to that. But in the presence of that PKC delta inhibitor, the interesting thing was that there was still a strong neutrophil migratory response that was observed in the murine cells, which was very different in terms of responses than the human donor cells themselves. And when we do a side-by-side comparison at just the 60-minute time point, it becomes even starker to really see that although there's a difference in terms of, maximal number of neutrophils that migrate in response to a pro-inflammatory cytokine, the key here is the anti-inflammatory treatment. While the human cells, the neutrophil migration went down significantly. However, in the murine lung microvasculature, there was still a larger number of neutrophils that migrated across. So not only can we really see a species-dependent, effect here, but then again, when it becomes to immune cell migration, those species-dependent effects really, come into focus. So in the remaining time that I have, I'd like to talk a little bit more about the toxicology treatment that we have. Also, we've incubated our lung epithelium with different levels of total particulate matter, which is a drug that can induce a loss of cell viability at higher concentrations. This is a Presto blue staining, but you can see as the increase in total particulate matter, we get a loss of viability. And we can also look in response to that drug treatment to look at reactive oxygen species expression, and also to see, the timing of this effect as well. In this case, when we incubate our cells, our lung epithelial cells with total particulate matter, if we're looking at 24 hours, one thing that we observed was that there was a resolution of the ROS staining. But if we look at 4 hours post-treatment, the interesting thing is that we really picked up a strong reactive oxygen species signaling with our 10 nanogram per mils of total particulate matter. And that particular ROS was resolved within that 24-hour period. So it lets us to, this platform allows us to do those time courses to really address that. And then we can also look at apoptosis, cell viability analysis, in this case with tunnel staining, showing less, loss of viability compared to higher concentrations. And then finally, we can also look at epithelial permeability, both at short and at longer periods of exposure with fluorescent tracers to look at how the lung epithelium is influenced in the presence of drug treatments. So in conclusion, our SynALI summary models that we can have co-cultures or we can have tricultures where immune cells are incorporated in using primary cells and cell lines. And we can use this for toxicology analysis, as well as to identify different influences on pro-inflammatory responses and responses to anti-inflammatories. We have a wide range of chips and sizes that are available. And we are located in Huntsville, Alabama, here at the Hudson Alpha Institute for Technology. Thank you again for your time, and I'll answer any questions in the remaining.
Thanks so much, Deborah. We do have about a minute, so there is one question in the chat that you can answer now. In terms of the endothelial cell responses in murine versus human, are there any different endothelial cell subtypes that could account for some differences in the species response?
That's a great point. In the study that I was referring to, they were definitely taking from the microvasculature. So they were looking at, specific sized vessels from the donors themselves. So the vessel size was very key in terms of the isolation of those cells. But we definitely see differential responses if we are looking at a HUVEC-based vessel, or if we were looking at cells that were taken from like an aortic for a cardiac model. So the vessel size at harvest definitely impacts the responses that we see, not only to just the pro-inflammatory cytokines, but also to the anti-inflammatories as well.
Awesome. Thanks, Deborah.
Hi, everyone. I'm Ashley from Draper in Cambridge, Massachusetts. Today, I'm going to speak with you about our high fidelity, high throughput, BSL-3 compatible human lung MPS model that we've taken advantage of to evaluate SARS-CoV-2 and screen associated therapeutics. So the COVID-19 pandemic posed some unique challenges and opportunities, which we're all well aware of at this point. Although there were highly efficacious and safe vaccines developed and deployed in record time that turned the tide of the pandemic, there has remained a need for the identification of and development of additional therapeutics to curb the wave of, to be used to curb the wave of infected individuals requiring hospitalization and reduce adverse effects, as there will always be individuals in the population who remain unvaccinated or who have breakthrough cases as new variants emerge. And a lot of times, time and resources were spent in search of these therapeutics that could blunt the response that individuals were having to SARS-CoV-2. And this took advantage of more simplistic cell culture systems and in vitro platforms. Only a few core candidates emerged. And so this has presented an opportunity for microphysiological systems to jump in line here and hopefully serve as a human relevant, more predictive preclinical tool to allow us that can be rapidly deployed and allow us to address pandemics and future emerging infectious diseases, such that we can quickly identify potential therapeutic candidates. So that's really what motivated Draper here. And prior to the pandemic, we had already developed a lung MPS system to study respiratory infectious diseases under 2 DARPA programs, Prophecy and Prepare. And we pivoted and modified this model to be adapted it for use in a BSL-3 environment to study infectious mechanisms of infection and evaluate therapeutic candidates to high priority pathogens like SARS-CoV-2. We developed a BSL-3 high containment capability along the way, where we have a laboratory of our own, in addition to individuals in-house at Draper with extensive deep training in high containment research. And since Draper can perform secure as well as select agent work, we're sort of uniquely positioned to look at certain pathogens utilizing our technologies in-house. Since this time, we've further matured our airway model, including additional immune components and sort of advancing on that front through collaborations and sponsorship by BARDA. And this technology has become our flagship microphysiological system model for our high containment research and medical countermeasure screening. So the technology, our MPS technology is known as Predict96. And its name is derived from the fact that our platform has 96 individual microfluidic bilayer tissue models specifically human tissue models, arrayed in a form factor that's akin to a standard SBS 384 well plate, where 4 individual wells of that 384 well plate correspond to an individual tissue model unit. They are bilayer devices, which means they have 2 chambers fanned by a semi-permeable membrane that can be functionalized with extracellular matrix and populated with cells. The top chamber has 2 ports of entry, as well as the bottom chamber. Cells can be populated on either side of this semi-permeable membrane, and they are subjected to fluid flow utilizing our precision microfluidic pumping lid. This is a sterile lid that is custom designed to sit intimately on top of the plate. Within that lid, we have 192 durable, precise micropumps that recirculate, that move fluid via pneumatic control. And move the fluid in one device around to create a hydrostatic pressure differential, and it drives fluid flow, excuse me, through our microfluidic tissue units. The stainless steel sippers that allow for the recirculation of fluid flow double as electrodes and permit integrated real-time sensing. So utilizing our custom software, and which is all Bluetooth controlled for remote data collection, we can just, through the click of a button, have multiplex data set pulled out for measuring things like barrier function via tear, oxygen, etc. So we can easily collect data in real time during the lifetime of our tissue's growth and development in culture. The materials of our platform are comprised of low absorptive materials, things like COC, such that we can evaluate compounds and have less loss. The technology Predict96 is highly compact in nature, so you can fit a number of these individual systems into a single incubator, which gives you up to about 3,000 individual tissues or organs in a single incubator at a given time. The form factor of this allows for it to interface with standard laboratory equipment, such as liquid handling robots or multiplexed pipettes. This allows for high throughput operation and automated workflows. The system is compatible with all types of omics analysis. The material can be taken out of our platform, both cellular material as well as supernatant and media. And there's an optically clear, our microfluidic stack is optically clear, such that you can do real-time in situ live and terminal imaging using any confocal or high throughput imaging modality. There's a specific version of our Predict96 technology referred to as ALI, or air liquid interface, that supports the development of our airway model. This has a basal microfluidic access port for fluid shear stress to be applied, at an open apical top, which allows for cell introduction. So we introduce basal progenitor cells onto that semi-permeable membrane. They're differentiated in situ and grown and matured under air liquid interface to generate tissue that has architecture, structure, and function that resembles that of the human in vivo lung. Here are just some examples of some of the key predominant cell types present in our model, ratios to be expected. The basal progenitor goblet, ciliated, and club cells. In addition to structural characterization, we do a number of functional types of characterization, including detection of mucus production, secretion of proteins like CCSP. We measure ciliary beat frequency and measure things like barrier function. The cell types that we source can be commercially derived, but we also have a special relationship with MGH, where we source cells via research bronchoscopy through collaboration with a couple clinicians in the pulmonary critical care division. This allows us to donor match airway epithelial cells to immune cell types for co-culture, as well as derive cells from very specific human populations with unique comorbidities. And this is something that we are spending some time digging into the weeds on, is how do individual human populations respond to certain pathogens and other threats. This is just a sampling of what the throughput really affords you with our system. It allows you to tune a lot of knobs depending on what you're interested in looking at. You might be looking at a compound at a range of doses, doing initial sweeps of different MOIs to understand how your model is responding to challenge with a particular pathogen. We can look at multiple donors on a given plate. And all of the standard molecular and biological assays are compatible with our system. And just some standard ones we use on the regular are things like, in situ imaging via immunofluorescence, TIR, as I mentioned. We do Luminex profiling, as well as RT-PCR, among other types of characterization, cytotoxicity, etc. So utilizing this platform, we went on to challenge it with SARS-CoV-2. This is a very robust model at this point. We've evaluated far more than 4 donors, but you can see here just a sampling of 4 donors that are all robustly replicating SARS-CoV-2 as measured by the viral copy number over 6 days post-infection. And in addition to RT-PCR, we do a range of other types of assays to assess viral growth kinetics, including plaque and TCID50 to correlate our data. We don't only look at viral growth kinetics over time, but we look at the host response, as I mentioned, through Luminex, IF, various measurements of cellular toxicity, and we, as I mentioned, do a range of omics. So transcriptomics is something that we do a little more frequently. And we've performed this work enough at this point that we have, as I mentioned, a very robust model. So you're seeing CV values under 15% between plates and within a plate, looking at different devices conditions. And this is all, CV values derived from data sets that are viral infection kinetics using RT-PCR. We have robust viral replication of various variants of SARS-CoV-2 across multiple donors. Here's a sampling of 3 donors, and we're looking at a terminal time point. This is an immunofluorescent imaging of SARS-CoV-2 nucleoprotein, both Delta and Omicron, at 6 days post-infection, and we're able to see, at least at this single time point, we do this imaging at different time points depending on the questions we have for a given study, but we can capture differential infection kinetics through a variety of means. Here is viral and copy number data. On the left-hand side, you can see that we're looking at a range of individual variants of SARS-CoV-2. You see unique infection kinetic profiles that are reminiscent of information that we know about Omicron having a more rapid replication timeline. So we see higher viral loads of Omicron at 2 days post-infection, and for the most part, things catch up, but generally Omicron and Delta loads are higher than Washington. And then if you look at the right-hand side here, we just blow out and look at Omicron alone. We can look at various donors' response to it, and we see unique donor profiles as well. So our platform and capability here is both donor and viral variant agnostic. And in order to grow and culture each of these donors, we don't alter any of our culture parameters. The chemical and biophysical environment is the same. So it's a universal protocol, and each donor and variant has been successfully implemented on its first attempt. Following, I just want to move on to some of our antiviral screening work. So following viral infection, which occurs apically on the tissue with a 2-hour incubation, we wash the virus away. And we can introduce the antivirals at a range of time points, but the data you're going to see next is 2 hours post-exposure to the pathogen. And it's introduced in the basal media, and it remains in circulation underneath the tissue. Here's an example of a plate map with 6 different compounds with sufficient controls, but of course we can put a number more, a greater number, I should say, of compounds on a given plate if one doesn't seek to have as many replicates or as many doses. So it comes down to your situation at hand. We've screened a whole range of antiviral compounds, that being FDA EAU approved, as well as precursor compounds and candidates that emerged early in other in vitro screens as potential candidates that later dropped out in subsequent clinical trials. We see a blunting of viral replication with compounds that we all know also well. So nirmatrelvir, this is the active component of Paxlovid, remdesivir, and molnupiravir. We see that a precursor to the Paxlovid is also efficacious, and the calpeptin, however, was not. So this aligns with some of the findings earlier on. And we see nice differential response. Although we can't make an in vitro and vivo correlation statement here necessarily, we do have data that tracks with clinical outcome where we see the greatest blunting of viral replication with the nirmatrelvir, slightly less with remdesivir, and then slightly less with molnupiravir. It's important to note that this is looking at one flavor of data. We look at a lot of different types of data with our individual studies to go well beyond this. We've continued to evaluate compounds that later were identified as false positives and dropped out in clinical trials, that being amidiaquin and fluvoxamine. So we've had all these assessed on individual plates, and we're able to confirm that we see blunting with the active component of Paxlovid, but not with these other compounds. So our platform is able to identify.
You do just have about 1 minute left. Just to
no problem. Yeah, so this is just some of the details of the work that we've done at BSL-3 and high containment with our staff, and the number of donors are 12 plus and growing, and many variants under our belt. And just to summarize, we have this airway platform that supports robust SARS-CoV-2 infection across all variants, and we are able to replicate clinical correlation to known therapeutics and false positives, and we're underway in advancing our airway model such that it has scaled manufacturing more types of real-time sensing and more advanced immune biology. Thank you all for having me today.
So hi, good afternoon. I'm happy you all guys stuck around for me in the final talk of today. So my name is Iris. I'm a scientist at Mimetas. And today I want to talk to you about our platform, which is the organoplate, and how it can help you in your lung-on-a-chip research. But first, let's talk a tiny bit about Mimetas. So what do? We're actually a biotech company focusing on organ-on-a-chip modeling. And we're really trying to develop human tissue models that are relevant really for new drug screening possibilities. And so these tissue models can be very broad. They can range from very simple barrier tissues, where we just have monocultures in, for instance, a 3-lane setup, or we can scale that up and make that very complex, adding different cell types together, for instance, looking at stroma co-cultures, but also interactions with vasculature or even immune migration. And this is just a couple of the feeds actually that we as a company focus on. So basically, what I want to talk to you about is our platform. This is the organoplate, and I have a short video to show you to highlight what it actually is. So the organoplate is a standard 384 well platform. However, we modified the bottom so it contains 64 microfluidic chips. And as you can see here, if we zoom in one of those chips, we have a central chamber and 2 channels on the side of it. And these are separated by tiny ridges that we call phase guides that prevent fluids from overflowing in the adjacent channels. What we can do, we can fill this central chamber with an ECM, an extracellular matrix. And this can be anything ranging from collagen to fibrin to hydrogels. And these ECMs can either be empty or they can be filled with cells. Next up, we can fill the side channels with cell suspension, for instance, endothelial cells, to allow culture of endothelial vessels. And what's beautiful here is this is actually a membrane-free platform. So there are no artificial membranes between the extracellular matrix and the cells we're adding next to that. What we then do is we put our platform on a rocker. This is basically just a tilting platform to enable gravitational flow through the system. And therefore, we get a little bit of shear stress and allow the endothelial cells to grow. So what I'm showing you here is, again, a little bit of an overview of this organoplate graft. You can see it on the right. And this is one of our products. Next to that, we have a 2-lane organoplate platform, a 3-lane, and also some custom models. So what I today want to focus on for the lung on the chip modeling is actually the graft and the organoplate air. So here, just to highlight it a little bit more, we can see that one chip is actually consistent of 6 adjacent wells. And if we zoom in on that middle chamber, for the lung on the chip modeling, here we can actually add a mixture of, for instance, endothelial cells and stromal cells to allow the formation of a vascular network. In this sense, we can create a very complex co-culture by adding lung epithelial cells on top. So if you have a look at the side views, that's actually shown here. We usually first culture the cells submerged, so we just add medium, so they allow a little bit of expansion and proliferation. But after the removal of medium, we have a lung co-culture and an air-liquid interface. So what's nice about this is that the vasculature that we're developing in the central is actually completely perfusible. So on the bottom right, we have added a fixed dextran dye to one of the channels, and you can see the entire network basically lighting up with all these interconnected vessels. On the left side here, I'm showing you a phase contrast image where you can see the culture basically at its mature state. So again, in this middle big chamber, we have the stromal tissue with the vasculature. On the side, we have additional vasculature tubes. And then on the top, we have the lung epithelium. So what we can do with the lung epithelium, we can either add small area epithelium or alveolar epithelium, or whatever a client is very interested in. And if we zoom in into the central chamber and do some stainings, this is what we can see. So for the small area epithelium on the left side, we have, for instance, marked the vasculature here by CD31 in green. And in pink, we have the basal cells marked by cytokeratin 5. And also we can indicate we can get a fully polarized epithelium just by this setup with both the ciliated cells and also the mucus producing cells. And the same actually applies for the alveolar epithelium, where we on the right side have some markers for caveolin 1, but also BCA3, just indicating that we have all the relevant cell types that you would want to have in a lung on a chip model. So actually, this is the part about the organoplate graft. We can also model the lung in a little bit more of a different structure, actually in a cylindrical tubule. So here we still have this big central chamber. However, now we created a hole in the middle, and that actually allows the epithelial sites to grow in a cylinder. Again, if we move medium that is added on top, we can actually culture these cells on the side of the ECM at an air-liquid interface. And here we can have a little look at how these cultures develop over time. So we get this fully polarized epithelium again, really nicely showing the basal cells in red, more at the bottom side. And at the apical side, we see the beta-4 tubulin indicating the ciliated cells marked in green. And this model also nicely, we have a little video here to show that we have the beating cilia in there. And these cilia also actually develop over time, and we see more development the longer we keep the place in culture. So these cultures can be kept stable for up to 35 days to allow the full differentiation. And then we can use them for a variety of things, depending on the disease a client is interested in. So I have an example here where we're having a look at goblet cell hyperplasia. The images I'm showing here are on the top, just a DNA stain, again, of this cylindrical model. And we did some exposures with both EGF and IL-13 to get these goblet cells activated. On the bottom, we have the mucus staining in green. And what you can see is that actually in the unexposed chips, we don't have a lot of goblet cells, don't have a lot of mucus. But if we add a couple of exposures, we can actually see them appear, and they significantly increase in expression. And on the right side, we actually quantify that as well, just looking at the signal intensity of the two different stains that we have. And we can clearly see a difference between the unexposed chips and the exposed chips with EGF and IL-13. And of course, this dose is different. This effect is different for different doses. So what we want to show today is that on one hand, we can successfully culture both the small area and or the alveolar array in the organoplate graft under air-liquid phase conditions. And on the other hand, we have these cylindrical bronchoepithelial cultures, but both sets can show the fully differentiated set of lung epithelial cells that we're actually interested in. So we have both basal cells, ciliated cells, and the secretory cells. And this is a very customizable platform. So we can use it for tox studies or for exposures. And what we really want to look forward to in the future is actually to look at certain exposure measures, for instance, through air, for aerosol exposures, and also have a little look at other airway remodeling readouts that we can apply to our current platform. And of course, what's very nice, because we have this vasculature that's already integrated, it would be very interesting to add an immune component to look at the interactions of then 4 different cell types into the same system. So how do we collaborate? Usually with researchers, we have a couple of different project types, really for more simplistic models, for instance, monocultures or just empty plates. You can always reach out to us, but in terms of the more complex, let's say lung-on-a-chip model systems, where we can really customize basically the projects to what a client needs, we have a fee-for-service model where we usually co-optimize models and readouts. And what the really big aim, of course, is to use our models for actual drug discovery pipelines. So that's really where we're hoping to get also in the future with, of course, using the MPS systems to get rid of all those animal testings and get more relevant models onto the market with human tissues. So a little recap again, who are we? So Mimetas, we really focus on the human tissue models, already working on that for 10 years with organ-on-a-chip technology with the organoplate platform. And there we have the different modalities with either 2 or 3 lanes or this big graft chamber in the center. And I really like to acknowledge, so the data that was generated here for the cylindrical model was actually with Japan Tobacco. So I want to acknowledge their collaboration together. And if you have any questions, let me know.
Thanks so much, Iris. And let me just check the chat. There aren't any questions in there at this time, but let's give it a couple seconds while people are thinking. All right, well, okay, we have a question just coming in from Peter. Please, can you make some comment on the relative shear stress achieved in your system versus perfused organ-on-a-chip systems and how that impacts cell differentiation and characteristics reflective of cells in vivo?
Yes, thank you. That's a very good question, actually. So as I mentioned, what we usually put the plates on is this rocking platform, and we can do that at different settings. So indeed, we induce a bidirectional flow because the platform goes back and forth. But depending on the orientation that you put the plate on, you can allow different kind of shear stresses, also dependent on the angle that we put the plate on and the speed at which we change. So for different endothelial cells, for instance, you can optimize that. So we have different shear stresses that are applied to the system. So this is something you can also basically customize depending on what kind of model you're working with. But it's not indeed, and the thing is, indeed, we don't use any pumps, and that also makes the system very easy of use, actually.
That's great. Thanks so much.
Well, thank you very much, Lauren. I think there was some great questions there, and it looks like there's some interesting synergies, but also the diversity of what we're seeing from these different companies is actually incredible. So I think our search towards progressing towards being able to make this a more standard approach and get a more human relevant toxicology and efficacy testing I think is really moving forward greatly. I don't want to hog the space, so are there any questions or anything that people want to discuss from the audience, or please go ahead and question the panel.
Clive, I think there are a few questions in the Q&A, so I can definitely read those out if can't see them there.
I'm not in the right place to look at them. I can open them up and have a look.
That's okay. I can totally read them out. So I think this question is directed to Ashley from Draper. The question says, I might miss it. What is the pore size of the membrane in your setup? Also, have you tried the chip with new modalities like immune cells gaugers?
Hi there. The pore diameter that we generally use for this model is 0.4 microns. The pore density is another feature of the membrane that we think a lot about. But we have the ability to utilize membranes with different pore diameters to suit different models that even go beyond the lung model that Draper has. And then, apologies, can you repeat the second question? To evaluate different modalities?
Yeah, so the question says, have you tried the chip with new modalities like immune cell engagers?
Immune cell engagers. I'm not quite sure what is meant by that specific question, but if someone wants to expand a little bit more just to clarify it for me, maybe more than happy to give it a go.
Yeah, maybe if the individual who asked this question, we'll give them some time to maybe clarify. There is another question for all of the speakers, so maybe I can ask that one now while the individual just clarifies. So the question says, thanks to all speakers for the great session. I have a general question. Internal validation can be sufficient for internal decision-making, but formal validation and qualification for regulatory acceptance is resource-intensive and time-demanding. Can you comment on how far along are lung MPS on these processes and what you see as biggest challenges in the process? Clive.
Definitely got an answer here. We actually don't know how we're going to be doing this. The big problem is that if we follow the ECVAM route, it's going to take years. Okay, and it's not actually appropriate to have this one method that we then follow for multiple labs, which stops innovation, and also it doesn't actually relate to how different technologies perform. So we are engaging with regulatory agencies to ask them about how we're going to deal with this very question. And we still haven't got necessarily all the answers, but in terms of advice we're getting back, it includes things like, can you demonstrate that you have reproducibility and repeatability? So that's like on the same day with the same set of drug candidates or drug standard molecules. Reproducibility, can you do them with different on different batches and with different batches of your cells? The best way of really doing this is to look at the paper by Van der Salm, which is actually stopping us from comparing against animal data, because we've so often gone, oh, hang on, we've got animal in vivo data, we're getting different results with our human in vitro. Oh, it means the in vitro studies failed. But actually, we've just ignored the fact that we're dealing with two species. So we've got four things going on. We've got in vitro, in vivo, and two different species. And we're probably finding that it's the species difference. And we should expect to have some amount of failure in this area, because the animal studies don't often replicate what we're having in the clinic. Because if there was, we would have no clinical failures for safety or efficacy. So we really need, so the answer to this is actually a bit more complicated. We actually need some guidance as to how we're going to do this. And it really appears to be, I think Emulate did a really good paper with their drug-induced liver injury at the end of last year. Those types of things where we've got defined chemicals, which we understand what they're going to do in the human or in the patient, is really where we're going to move forward. And I guess that another great way is to bang on the door of IQ Consortium and say, please give us some of your drugs which have failed at different points and tell us why they failed. So a direct question there. So I didn't answer your question, but I think it's big in that, and I'll pass that over to other people who may have a more direct answer.
Yeah, I totally agree with everything Clive said. I think it's a very nuanced question and answer, and it's very complex. I think we're having this conversation across all the MPS fields currently, and it's very much based on the application and the requirement. So you can't really have that conversation in isolation or as a sort of broad topic. It's very, very specific. And I think that as Clive sort of mentioned, is that, you know, there are so many in vitro models out there right now, which, you know, all are very useful, but perhaps for different reasons or for different applications. And it's just having that standardization that he mentioned that's really crucial right now. And I think there was mention in the chat about standardization of, for example, cell markers. You know, characterization of the models, I think, is really important at this point, to really ensure that we're all sort of going for the same goal, and we all have the same standards. So I think before we can say about the regulatory side, we actually have to get to a point where you can characterize your models to standards, and that's really, really important. So I think that's where the IQ Consortium is crucial, for sure.
And if there's anyone else, oh yeah, Megan, go ahead.
Sorry, yeah, just to touch on that, I think I completely agree with what's already been said, but also there are examples where lung MPS models have actually been used for regulatory approval previously, coming back to cystic fibrosis, where the drug for CF was actually approved for rare mutations based solely on in vitro data. So I think there is a way forward, which has been shown in the past, but completely agree that we need to kind of identify what an ideal model looks like and how we can all sort of benchmark those standards to really facilitate that drug discovery process and the regulatory approval as well.
Do any other individuals, any of our speakers have anything else to say on that topic? Maybe just
I think that Pete Gaskin's comment appears to be related into this. Pete, did you want to speak a little bit more about what you've said there?
I don't know.
Can we
Yeah, I
give
don't
him
think
some?
Pete will be able to turn the mic on himself. But if the group
Yeah, he's trying to. It'd be great if we can get Pete to speak here, because I think that's a very relevant question.
Yes.
I will actually jump in here real quick. This is Megan from the 3Rs Collaborative. Oh, that's the wrong camera. One moment. So I'll just speak real quick. And so the question or his comment was if we need a new and more regular forum for industry, including CROs and service providers, to interact with regulatory authorities. And I can say that the 3Rs Collaborative is in the process. We have put in an application for a consortium agreement with CDER at the FDA, which would allow us, our organizations, our member companies, to interact more directly with the FDA. And I believe that the IQMPS also has a consortium agreement with CDAR as well, which allows them to interact with the FDA. I can't speak to European agencies, but at least CDAR at the FDA has such forums and is very interested in such interactions. There's also the iStand program in a variety of ways. You can actually apply already to give webinars to the FDA. So there are a number of forums for such interactions.
That's great. Thanks, Megan. And Peter, if you just wanted to, as Clive director, just expand on your question if there was more to elaborate.
Yeah, thanks for giving me a chance to speak. Yeah, I just wanted to make some comment really, because the IQMPS, for example, my understanding is that consortium doesn't allow CROs to join. It's only for industry. So whilst that consortium is great, and you know what that consortium has been doing is fantastic, you know, those of us who don't work in a pharma company, don't have a drug in a clinical trial, aren't able to engage in those forums. And you know, if we as an industry want to progress on this, you know, service providers who are running studies for clients need to be part of that party.
And I'll say, Peter, that you are always welcome to join our initiative. Although we are primarily the commercial providers, we are actually welcome to CROs, any type of service provider, as well as end users. And so you're always welcome to join our group and be part of us there. And as Charles